Welcome back, everyone, to the Deep Dive. Today, we're really going to get into the nitty-gritty of things, diving deep into a groundbreaking research article about, get this, a newly discovered neurological disorder in Weimaraners.
Really fascinating stuff.
Now, we're getting pretty technical here, kind of like neurologist-level stuff.
Yeah.
So if you're into myelin abnormalities, Lipofuscin-like inclusions, and even, get this, a connection to mitochondrial function, well, you're in for a treat.
It's groundbreaking research, really unveils a progressive neurological condition we've never seen before.
This deep dive is all based on this, super in-depth case report they just published.
Yeah.
And they go through everything, the clinical presentation, all the diagnostic tests they did.
Yeah.
And even what they found during the postmortem exam.
They start with this 5-year-old male Weimaraner, neutered, who came in for a neurological evaluation. The owner was reporting a 3-month history of ataxia in the pelvic limbs, some fecal incontinence and just lethargy.
So already some red flags popping up right there.
Definitely. And when they examined him, they found moderate paraparesis and ataxia, that proprioceptive kind, in the pelvic limbs. So initially they were thinking, okay, maybe it's myelopathy.
It makes sense.
But things got interesting when they did the imaging.
Oh, what'd they find?
Well, the spinal MRI, nothing, really. No significant abnormalities. So they shifted focus to the brain, and that's where they saw it pronounced cerebral parenchymal atrophy.
Wow, so the brain was actually shrinking.
Yeah, and they saw evidence of this in several places.
Like what?
Well, the interthalamic adhesion was, way smaller than it should be.
Okay.
The third ventricle was enlarged, and the subarachnoid spaces widened.
Interesting.
Plus, they found these areas in the cerebral cortex white matter that had this increased signal intensity.
So lots of structural changes in the brain.
Yep.
What about the cerebral spinal fluid analysis? Did that show anything?
Completely unremarkable.
Adding another layer to the puzzle.
Really is. Unfortunately, the dog's condition just kept getting worse over the next year and a half.
Oh, that's so sad.
He started falling more, the fecal incontinence worsened, and even signs of cognitive decline.
Oh, no. Poor guy.
The owners also reported decreased appetite, changes in his posture, and even these weird trance-like episodes.
It sounds like a really tough situation for everyone involved.
Absolutely.
Yeah.
And sadly, because of how severe the neurological dysfunction was becoming,
Yeah.
they decided to elect humane euthanasia.
I understand.
It was a tough call, but it allowed them to do a really thorough post-mortem examination.
And I imagine that provided some valuable insights.
Definitely.
So what did they find?
Well, one of the first things that jumped out at them were these autofluorescent granules.
Granules.
Yeah, they were inside cells of the cerebral and cerebellar cortex.
Interesting.
And they were really concentrated in the Purkinje cell and granule cell layers of the cerebellum.
Okay.
Now, you might think they only found these granules in the brain.
I was going to ask that.
But nope. They were scattered throughout the cerebral cortex gray matter, but absent in the neural retina.
Really?
Yeah, and in the optic nerve they were there.
Okay.
But they looked different morphologically speaking.
Interesting. So some regional variation.
Exactly.
Right.
Oh, and they also found these granules in the cardiac muscle.
In the heart?
Yep. Clustered in these linear arrays within the muscle fibers.
That's wild. So they were popping up all over the place, but not uniformly.
Right.
So what do these granules look like under the microscope?
Well, they were pretty striking, actually. Under a fluorescence microscope, they had these emission colors ranging from golden yellow to orange.
Oh, wow. That's pretty vivid.
Yeah, to really get a good look at the structure of these granules, they used transmission electron microscopy.
Getting down to the nano level.
Right.
So what'd they see?
Lots of interesting stuff.
Like what?
Well, they found these membrane-bound organelles filled with all sorts of different stuff. And these organelles were in various brain regions, plus the optic nerve.
So pretty widespread.
Yep, they saw them in the cerebellar cortex gray matter, cerebral cortical gray matter, cerebral cortical and cerebellar white matter tracts, and of course, the optic nerve.
So pretty much everywhere they looked.
Pretty much.
What was inside these organelles?
Well, it was a mix of things, like granular components with different electron densities, membranous components, and even lipid-like inclusions.
Wow, so a real hodgepodge of stuff.
Yeah, it was pretty remarkable.
So we've got these weird granules, and they're inside these even weirder organelles.
Yep, and it doesn't stop there.
Oh, no.
They also found abnormalities in the myelin sheaths.
Uh-oh, that's never good.
especially in the optic nerve.
What was going on there?
They saw all these like large gaps between the myelin layers.
So the myelin wasn't forming properly?
It looked like it.
Oh,
And
really?
get this, it seemed like abnormal myelin was like budding off from the axonal sheaths and then getting taken up by nearby cells.
Oh.
Yeah, and those cells were forming these structures with like whorls of membrane.
That's bizarre.
It was really strange.
So the myelin was messed up in the optic nerve. What about other parts of the brain?
They found similar abnormalities in the brain's white matter tracts.
Really?
Yeah, pretty much all the axons had these distinctive gaps between their myelin layers.
And we know myelin is, like, crucial for proper nerve function.
Absolutely.
So these abnormalities could definitely explain some of the neurological symptoms.
Right. They even saw degeneration in some axons.
Oh, no, so the axons were actually breaking down.
It looked that way. The axoplasm was being replaced by, like, myelin-derived membrane and membrane fragments, and it seemed like it was collapsing inward.
That's a pretty grim picture of what's happening at the cellular level.
Yeah, not good.
What about those inclusions in the cardiac muscle? Anything special about them?
Actually, yeah, those were particularly interesting.
Oh, so?
Well, they were all clustered in these areas that were packed with mitochondria, and they looked more uniform than the ones in the brain and optic nerve.
Okay.
They were made up of these parallel arrays of membrane-like stuff, and then on top of that, there were these patches of very electron-dense material.
So a different structure than the other inclusions.
Definitely.
It's amazing how they can see all this detail with electron microscopy.
It really is. It helps us understand just how complex this disorder is.
For sure.
But they didn't stop there. They wanted to know more about what these inclusions are made of, so they used immunohistochemistry.
Okay, so let's talk about those findings.
Yeah.
What did the immunohistochemistry reveal?
One of the biggest discoveries, and this is like huge,
Yeah.
the disease-related inclusions in the cerebral cortex, gray matter. They were labeled with an antibody that targets subunit C protein of mitochondrial ATP synthase.
Hold on, mitochondrial ATP synthase, isn't that involved in energy production?
Bingo.
So this disorder might not just be about structural abnormalities, but also about problems with how cells make energy.
Exactly. It's like this deeper level of dysfunction.
That's a pretty big deal.
It is.
What about the cerebellum? Did they see the same thing there?
Interestingly, no. In the cerebellar cortex, only a very small number of the inclusions were labeled with that antibody.
So some regional differences in how this protein is involved?
Right.
What about the heart muscle?
That one showed very strong immunolabeling with the same antibody.
Interesting.
And just to be sure, they also tested another mitochondrial protein,
Which one?
the subunit of mitochondrial synthase.
Okay.
But none of the inclusions in either the brain or the heart were labeled with that antibody.
So it seems like there's a very specific involvement of subunit C, of mitochondrial ATP synthase.
It really does.
It's like each new piece of information adds another clue, but we're still trying to figure out the whole picture.
Exactly. But on top of all this, they also found evidence of neuroinflammation in the brain.
Oh, so the immune system is involved as well.
It seems so.
Tell me more about this neuroinflammation. What did they observe?
Well, they saw pronounced astrocyte activation both in the cerebral cortical gray matter and the cerebellar cortex.
Okay.
And there was pronounced microglial activation in the cerebral cortex.
But not in the cerebellum.
Right.
Interesting.
And you might remember we talked about a similar disorder in Dalmatians.
Vaguely.
Yeah, well, that one was linked to the absence of a specific enzyme called CNPase.
Okay, now I remember. So did they look for CNPase in this Weimaraner?
They did, and get this, they found a similar lack of CNPase.
Wow, really?
Yeah, when they looked at brain tissue from a control dog, they saw tons of immunolabeling for CNPase.
But in the Weimaraner.
Nothing.
So there's a potential link between this new disorder and this known enzyme deficiency that affects myelin.
Exactly. And to really dig into this connection, they turned to genetic analysis.
Okay, let's talk genes. What did they find?
So they did whole genome sequencing on the dog's DNA.
Fancy.
And bam, they found this rare homozygous G to A transition.
What?
A change in the DNA sequence.
Okay.
It was at a specific spot, position 20,355,460 on chromosome 9.
Gotcha.
And this mutation, they predicted, would cause a THR42-MET missense mutation in CNPase.
Wait, so this mutation could explain why they weren't seeing any CNPase protein.
It's very likely.
How can they be so sure it's important?
Well, they did this thing called a blast-up query. And it showed that a threonine residue at that exact same position, 42, was basically the same in 100 different mammalian CNP ortholegs.
So across all these different species, that specific amino acid has stayed the same.
Yep.
Which means it must be pretty important for the protein to work, right?
Exactly.
So not just a random mutation?
Nope. This one seems to be a big deal.
And to top it off, it was super rare. They only found it in 3 out of like almost 8,000 alleles they analyzed. So this is getting really interesting.
It is, and it gets even better. You won't believe this, but while they were working on this case, they found another Weimaraner.
With the same mutation?
With the exact same homozygous mutation.
No way!
I know, right? What are the chances?
So 2 dogs with this super rare mutation, and they both have similar neurological problems.
Hold on, let me tell you about this second dog.
Okay, I'm listening.
This one initially came in because of bilateral hind limb weakness and, you guessed it, fecal incontinence.
Sounds familiar?
Right. And they ruled out degenerative myelopathy based on SOD1 genotyping.
So not the typical Weimaraner spinal cord problem?
Nope.
Okay, so what else was going on with this second dog?
Well, his story was pretty similar to the first one. The pelvic limb weakness started when he was 8 years old.
Okay.
He'd had progressive fecal incontinence for about a year. He also had ambulatory paraparesis and, get this, generalized whole body tremors.
Wow, poor guy.
Yeah, the tremors, those were a pre-existing condition, not related to this.
Okay.
And just like the first dog, his brain MRI showed significant, generalized brain atrophy.
So shrinking brain again.
Yep, and those areas of increased signal intensity in the cerebral cortex parenchyma, just like the first dog.
So even the imaging findings were similar.
Yeah, pretty striking, right?
Did they do a cerebrospinal fluid analysis on this dog too?
They did. It showed a slight increase in protein concentration, but the cellularity and cell types were normal.
So a little bit of a difference there?
Yeah.
Did they try to treat him?
Oh, they tried everything. Immunosuppressants, steroids.
Nothing worked.
Unfortunately, no. Nothing could stop the progression of the disease.
That's so frustrating.
It is.
And ultimately, they had to euthanize him as well.
Yeah, it's always tough when you see these progressive conditions that just don't respond to treatment.
Absolutely. But at least they were able to gather so much information from these 2 cases.
You're right. It's invaluable for understanding this new disorder.
So what do they do next? Do they try to figure out how common this mutation is in Weimaraners?
They did. They got their hands on 304 archived Weimaraner DNA samples.
That's a lot of samples.
It is. And guess what? They found 9 that were heterozygotes for that G to A transition.
So these dogs had 1 copy of the normal gene and 1 copy of the mutated one.
Exactly.
What happened to those dogs?
Well, one of them was only 3 years old and didn't have any symptoms.
Interesting.
But the other 8, their ages ranged from 8.8 to 13 years old.
Okay.
And guess what? They all had slowly progressive hind leg weakness and other signs of neurodegenerative disease.
So even having just 1 copy of the mutation could cause problems.
It seems so.
Wow.
And just to be sure, they tested all these dogs for the SOD1 variant.
The one linked to degenerative myelopathy.
Yep, and they were all negative for that.
So their neurological problems weren't from the typical Weimaraner spinal cord issue?
Right.
What about the rest of the samples?
The other 293? They were all homozygous for the normal CNP allele.
2 copies of the good gene.
Yep.
So even though this mutation is rare, it seems like it's a pretty big deal for Weimaraner health.
Definitely.
Did they calculate how common the mutation is in the breed?
They did. They estimated the frequency of the variant allele to be 0.021 in that collection of samples.
Okay, so that gives us an idea of how often it might pop up.
It does. And now, with all this data in hand, they could finally start to put the pieces of the puzzle together.
Okay, this is where it gets really good. What did they conclude about this new neurological disorder?
Well, that, my friend, is a story for another time. All right, so remember all that data we talked about in the last part?
Yeah, the clinical signs, all those weird microscopic findings, the whole CNPase thing, and that super rare mutation.
Right. Well, the researchers really did some impressive work putting it all together like a giant puzzle.
So what was their final diagnosis? What did they conclude was going on with these Weimaraners?
They think it's a lysosomal storage disease, probably similar to neuronal ceroid lipofuscinosis.
Unseal.
Yep, that's the one.
Okay, but for those of us who aren't, neurology experts, can you break that down a bit?
Right.
What exactly is a lysosomal storage disease?
Sure. So think of lysosomes like the recycling centers of the cell. They've got these powerful enzymes that break down all the cellular waste and junk.
Makes sense.
But in lysosomal storage diseases, those enzymes, they're either missing or they're not working right.
Uh-oh.
Yeah, so all that waste product, it just builds up inside the cells.
And that's bad.
Definitely. It makes the cells swell up, and eventually they can't function properly.
So in these Weimaraners, the lack of CNPase is causing this buildup of cellular waste.
Exactly, and that's probably leading to all those neurological symptoms we've been talking about.
So it's like the cells are drowning in their own garbage?
Kind of, yeah.
But you said this disorder is similar to NCL.
Right, because there's a twist.
What do you mean?
Well, remember all those myelin abnormalities we talked about?
Yeah, those are pretty striking.
Those aren't really typical of classic NCL.
So it's like NCL, but with a myelin problem on top of it.
Exactly. It suggests that this CNPase deficiency is impacting myelin in a way that's, well, unique.
Interesting. So there's like a variation on the theme. Have there been other cases of CNP deficiency in other animals?
Yeah, actually, there have been reports in mice, humans, and even other dog breeds.
So they were able to compare their findings to what's already known about CNP deficiency.
Yep, and that helps them understand this particular disorder in Weimaraners even better.
What about that THAR42 MET mutation they found? Where does that fit into all of this?
That mutation is probably the key to the whole thing.
How so?
Well, remember how they couldn't detect any CNPase protein in the affected dogs?
Yeah.
This mutation is most likely why.
You mean it's preventing the protein from being made?
Well, it's either that or it's making the protein so unstable that it gets broken down immediately.
So basically the protein's not doing its job.
Right, and we see the same thing happening in humans.
Really?
There's a human disorder caused by a different mutation in the same gene.
And it also results in a lack of CNPase?
Yep, almost completely absent.
Wow, so these specific spots in the CNPase gene, they're really important.
Definitely. Even a tiny change can have huge consequences.
It's amazing how much we're learning about genetics these days.
It is.
So remind me again, what does CNPase actually do?
It's got two main jobs. First, it's an enzyme that breaks down nucleoside 2'3" cyclic monophosphates.
Okay.
And its second job is related to myelin.
I knew it. Tell me more.
CNPase is a major part of myelin in the central nervous system.
So it helps build and maintain that protective sheath around nerve fibers.
Exactly. So if you don't have enough CNPase,
You're in trouble.
yeah, you're messing with both the myelin structure and the breakdown of these important molecules.
Double whammy.
Right. And that brings us to the big question.
$1,000,000 question.
How does all of this, the myelin problems, the lysosomal storage dysfunction, maybe even the mitochondrial involvement, how does it all lead to those specific neurological symptoms we saw in these dogs?
That's what I want to know. Break it down for me.
Well, the myelin abnormalities are probably the most direct link.
How so?
Remember all those gaps and disruptions we saw in the myelin sheath? Myelin acts like insulation for nerve fibers. It helps electrical signals travel quickly and smoothly.
Okay.
But when the myelin is damaged, those signals get all messed up.
And that causes problems with movement and coordination.
Exactly. That's likely why these dogs were having ataxia and weakness.
Makes sense. What about the other symptoms, like the fecal incontinence and cognitive decline?
Those are a bit trickier. They probably involve a combination of things.
Like what?
Well, the lysosomal storage dysfunction could be causing widespread cellular stress.
So it's not just the myelin that's affected.
Right, different brain regions could be struggling, and that could lead to a wider range of neurological issues.
And the mitochondria. Could they be playing a role too?
That's the burning question.
Okay, I'm all ears.
So we know those inclusions were labeled with the antibody for subunit C of mitochondrial ATP synthase.
Uh-huh.
Well, that suggests that there could be something wrong with how the mitochondria are working.
So this mutation in CNPase might be affecting the cell's power plants.
Exactly.
Wow, that's a lot to take in.
It is. And it makes sense when you think about it.
How so?
Myelin is really energy intensive. It needs a constant supply of ATP to stay healthy and do its job.
So if the mitochondria aren't working right,
Then the myelin problems could get even worse, leading to even more neurological dysfunction.
it's like a vicious cycle.
Exactly. And remember those abnormal myelin fragments?
Yeah.
Well, they could be getting taken up by other cells, clogging up the works, and making the lysosomal storage problems even worse.
It's amazing how everything's connected.
It really is. This case just shows how complex biological systems can be.
And how important it is to look at the whole picture, not just one piece of the puzzle.
Absolutely. So where do we go from here? That's the next question.
Okay, I'm ready. What are the next steps in understanding and treating this disorder?
Well, there's still a lot we don't know, but this research has given us a great starting point.
Yeah, it's like we've opened this door to a whole new world of understanding about this disorder, but there's still a long way to go.
Absolutely. And the first step is to really dig into that THOR42 mutation.
Okay.
We need to figure out exactly how it's messing with the CNPase protein.
You mean like, does it make the protein unstable, or maybe it can't get to where it needs to be in the cell?
Exactly. Maybe it's interfering with how it binds to other molecules. There are a lot of possibilities.
So we need to figure out the exact molecular mechanism.
Right. And for that, we can use things like cell culture and protein modeling.
So basically, we can recreate the mutation in the lab and see what happens.
Yep. We can see how it affects CNPase production, where it ends up in the cell, how active it is.
And we can use computer models to visualize how this mutation changes the protein structure.
Exactly.
And if we can understand how the mutation is disrupting CNPase, maybe we can find ways to fix it.
That's the hope, right? Like, if we find out the mutation is making the protein unstable, we could try to develop drugs that stabilize it or boost its production.
What if the problem is that it can't interact with other molecules properly?
Then maybe we could design drugs that mimic those interactions.
So there's a lot of potential for future research to lead to actual treatments.
Definitely.
You know, something else we need to figure out is the whole heterozygote thing.
Right. Those dogs with one copy of the normal gene and one copy of the mutated gene.
Yeah, some of them did develop neurological problems just later in life.
That's the big question. Is it just a milder, delayed form of the disorder?
Or is it something else entirely?
Exactly. Does having that one good copy of the gene offer some protection?
Or does it just buy them some time before the symptoms hit?
That's what we need to figure out.
So we need to follow those heterozygous dogs over time, see what happens to them.
Exactly. Longitudinal studies are key here.
We can track their neurological health, see if there's a pattern to how the disease progresses.
And from a practical standpoint, now that we know which gene is involved,
Right.
we can start testing dogs for this mutation.
That's huge for breeders.
Absolutely. They can screen their dogs and make informed choices about breeding.
To prevent puppies from being born with this disorder.
Exactly. So this research has a direct impact on the health of Weimaraners.
It's amazing how quickly we've gone from discovering this disorder to having the potential to prevent it.
It really is.
But this research goes beyond just Weimaraners, doesn't it?
Right. The fact that we're seeing similar CNPase deficiencies in other animals,
Like mice and humans.
it tells us that there are probably shared biological pathways at play.
So what we learned from these dogs could help us understand neurological disorders in other species.
Exactly. And maybe even lead to treatments for human diseases.
That's incredible. Studying dogs could actually help us humans.
It's the power of comparative medicine. You know, we're all more connected than we think.
This has been such an amazing deep dive. We've learned so much about this new disorder in Weimaraners.
It's a great example of how science can unravel the mysteries of disease.
And how important it is for researchers, veterinarians, and breeders to work together.
Absolutely. By combining our knowledge and expertise, we can make a real difference for both animals and humans.
Well, that's all the time we have for today. Thanks for joining us on this incredible journey into the world of canine neurology.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.